Hello everyone! Today we are going to learn about the Bradford Assay. The Bradford Assay was developed by Marion McKinley Bradford in 1976 as a method that could be used to help find the concentration of a protein in a sample. But why would you want to find the concentration of protein in a sample? That's a great question. You would want to find the concentration of a protein in a sample in order to study its expression and find out how much there is. For example, let's just say you were putting a cell under different stressful conditions and wanted to study whether the expression of a certain protein increased or decreased in that cell. You would need a quantitative assay that could track the protein's concentration with respect to time. In other words, a way you could track how much the protein was present. The Bradford Assay would save the day. But how do you do the Bradford Assay? First, a little understanding of an important reagent is needed. The Bradford Assay contains a chemical called Kumasi blue dye, and based on how much proteins are present, it will change colors from a brown color to a blue color. This is because certain amino acids will react with the dye to absorb blue light. In other words, the more blue the sample is, the greater the concentration of proteins that will be present. The Bradford Assay has saved the day. But what actually happened? Now we need to take a closer look at the protocol. In order to do the Bradford Assay, you will need your handy-dandy set of materials: cuvettes, parafilm, PBS, pipettes and tips, protein standards ranging between 0.125 to 2.0 milligrams per milliliter, your protein of interest, and a spectrophotometer. And the star of the show, the Bradford Assay reagent. Now you are ready to find the concentration of that protein you have been wondering about for so long. Let's begin. First, you want to prepare your protein samples in cuvettes. It is helpful to take good pre-lab notes and have a plan laid out in advance so that you know what you are doing. Look at this example. Here, we list how much Bradford Assay reagent and protein standard to put into each cuvette, as well as the proper concentrations if necessary. As we said before, the Bradford reagent is what will change colors in order to indicate the concentration of the protein present. At 595 nanometers, the spectrophotometer will read how blue the Bradford Assay is to quantitatively tell us how much of the protein we have. The protein standard is what will attach to the Bradford reagent. Nifty, huh? With a plan like this, you are ready to start working. And first, you want to pipette 1 milliliter of the Bradford Assay reagent into all of your cuvettes. Make sure to use new pipette tips every time you pipette and dispose of the old ones into biosafety bags. After that, you will want to add the correct concentration of protein standard to 7 of your Bradford Assay reagent cuvettes, as you laid out in the table you planned. Make sure to set one cuvette as your control and only add PBS to the Bradford Assay reagent, and to use another cuvette to contain your protein of interest. Parafilm your samples and invert them gently. Are you with us? Now you can take a break as you incubate your samples for 15 minutes at room temperature. In this time, you can think of how amazing Marion McKinley Bradford is and reflect on how she got the University of Georgia Distinction Award just last year. A great day for the Bradford Assay. At this point, set up your spectrophotometer to 595 nanometers in order to prepare to analyze the samples. But what is a spectrophotometer? Another excellent question indeed. Invented in 1940, a spectrophotometer is a machine used to quantitatively measure the reflection of a material with the function of its color displayed as a wavelength. Molecules have specific makeups, and because of them, they are able to absorb specific wavelengths of light. Spectrophotometers have a lamp that strike light to a diffracting gradient so that the light is separated to particular wavelengths, and these wavelengths interact with the molecules in the sample. A detector detects just how much the light interacts with the sample, and it displays its information in absorbance or how much light the sample absorbs. Remember how we spoke about Kumasi blue dye that changes color from brown to blue so that we could tell when there is more protein present in our sample? Well, the Kumasi blue dye reacts with amino acids, which make up proteins. The more amino acids there are, the more proteins there are, and the Kumasi blue dye will react with the amino acids. You could then use this to measure the concentration of the protein of interest within the sample with the use of the spectrophotometer. It all makes sense now. Hooray for the Bradford Assay! Time to get to work. First, blank the spectrophotometer using your first cuvette that has PBS. Your blank will be a point of comparison to look at your other samples alongside. Be sure to have on your gloves so that oils on your skin will not disrupt light from the spectrophotometer. Use a Kim wipe to make sure that your cuvettes are extra clean. Next, measure the absorbances of each of your prepared samples, and finally, your protein of interest. Be sure to record your values in a notebook for safekeeping. But what do we do with all this information? Let's take a look at a sample set of data and explore the process of data analysis together. A big Bradford yay! Look here at the samples and absorbances. They all contribute to an amazing story, which is possible to tell graphically through the use of Microsoft Excel. Create a new Excel file and put in your data. Insert a graph, and it will be possible to create a Bradford Assay curve. We will display our sample curve below. What does Bradford say? Well, the y-axis indicates the levels of absorbance in nanometers, whereas the x-axis indicates the concentration of protein in milligrams per milliliter. By pressing chart elements, pressing trend line, and going to more options, it is possible to add a slope equation to your chart by pressing display equation on chart. The idea behind the chart is the slope formula y equals mx plus b. Y is the absorbance, m is the slope, and x is the concentration of your protein which you solve for. To find the concentration of your protein, take your linear equation the chart generated for you. In our case, y equals 0.553x plus 0.0036. Next, identify the absorbance of your protein of interest from your lab notes. In our case, desalted aleuate, which was 0.28. After that, substitute that value into your absorbance value y and solve for x, your concentration. X, or our protein of interest concentration, is equal to 0.28. That means that our protein of interest is present. A big Bradford party! When Marion McKinley Bradford successfully did this, it was certainly a celebration for her too. And her paper on this procedure is one of the most cited scholarly articles of all time. Hip hip assay! Are you ready to find the concentration of your protein of interest? Now that you're properly prepared, find a good day to do the assay and get on your way. Until next time, have fun learning!